Good morning, students. I myself, Dr. V.B.Kadam, Associate Professor, Department of Botany, KTHM College, Nasik. Students, today we are going to learn the chapter Respiration and Lipid Metabolism, and today's topic is fatty acid biosynthesis. Plants produce the majority of lipids in the world. These lipids are the main source of calories and essential fatty acids for men and animals. Plants synthesize a huge variety of fatty acids. Only a few are major and common constituents like palmitic, oleic, linoleic, and linolenic acids. Like other eukaryotes, lipids are necessary for the biogenesis of cell membranes as signal molecules and especially as a source of carbon and energy. In plants, carbon, energy, and reducing power are needed for fatty acid biosynthesis, derived from photosynthesis in chloroplasts. Fatty acid biosynthesis in plants occurs in the chloroplast of green tissues and in the plastid of non-photosynthetic tissues and not in the cytosol as in the animal cell. The de novo synthesis is located in the stroma. Plant mitochondria are capable of limited fatty acid synthesis. All the fats are lipids, but all the lipids are not fats because some lipids are waxes, cutin, suberin, or even steroids, which differ from fats. Fats are simple lipids that are saponifiable esters of long chain fatty acids and glycerol. These are heterogeneous organic substances composed of carbon, hydrogen, and oxygen. All the fats are soluble in organic solvents like acetone, alcohol, chloroform, benzene, and ether, and soluble in water. One of the constituents of fats is glycerol, which is trihydric alcohol. When all the three hydroxyl group of glycerol are esterified with the fatty acids, the resulting triglycerides are known as neutral fats, conveniently referred as fats. Important properties of fats. These are soluble in organic solvent and insoluble in water. The oxygen content of fats is poor as compared to carbon. Fats are triglycerides, that means all the three alcoholic groups of glycerol are esterified. Most of the fats are in the liquid state in plants at ordinary temperature and are termed as oils. When fats and oils are heated with alkali, free fatty acids and glycerol are liberated, and this process is known as saponification. Distribution of fats in plants. The fats and oils are found in small amount in leaves, stems, and roots. That means approximately 0.5 to 2% the dry weight of the organ, whereas These are stored in higher amount of seeds, especially in endosperm, cotyledon or other storage tissues. For example, coconut fruits are rich in lauric acid, castor beans contain ricinoleic acid, etc. Generally, the fats is stored in specialized bodies called lipid bodies, spherosomes or oleosomes. Synthesis of fats. Fats and oils are generally synthesized in the cells in which they occurs. Because they cannot be diffused from cell to cell, it has long been suspected that the fats are synthesized at the expense of sugars translocated to these cells. The fat synthesis may conveniently be divided into three parts: the synthesis of fatty acids, the formation of glycerol, and combination of fatty acids and glycerol to form fats. Are the some structures of fatty acids: lauric acid, pyristic acid and palmitic acid. First, synthesis of fatty acids. Nearly all the long chain fatty acids contains an even number of carbon atoms. For example, lauric acid contains 12 carbon atoms, pyristic acid contains 14 carbon atoms, palmitic acid contains 16 carbon atoms, and stearic acid contains 18 carbon atoms, and so on. This suggests that these are built up by the repeated addition of carbon units. Further studies demonstrated that acetyl coenzyme A produced from carbohydrates during respiration provides the necessary carbon atoms for fatty acid synthesis. Later, it was shown that carbon dioxide was involved in the formation of a 3-carbon compound, malonyl coenzyme A, from acetyl coenzyme A, first time found by the scientist Klein in 1957. The reactions are as follows. Acetyl coenzyme A combines with bicarbonate ions plus one molecule of ATP with the magnesium ions and biotin to form malonyl coenzyme A and ADP and inorganic phosphate. This reaction requires the involvement of vitamin biotin, manganese ions, ATP and an enzyme carboxylase. Further steps in the synthesis of fatty acids are as follows. The acetyl coenzyme A get attached to an acyl carrier proteins. The acetyl coenzyme A acyl carrier proteins combines with acetyl S acyl carrier proteins to form acetyl S acyl carrier proteins. The malonyl coenzyme A reacts with reduced acyl carrier proteins and form malonyl derivatives of acyl carrier proteins, malonyl S acyl carrier proteins. The malonyl S acyl carrier proteins then condense with the primary acyl S acyl carrier proteins. The carbon dioxide is released and the condensation reaction leads to a chain of 4 carbon atoms. These 4 carbon atoms, acetoacyl S acyl carrier proteins, is then converted to a saturated fatty acids by 3 successive reactions as follows. Reduction dehydration, and again the reduction. Now in reduction, acyl carrier proteins plus NADPH to form alpha hydroxybutyryl S acyl carrier proteins and some molecules of NADP. After reduction in dehydration of alpha hydroxybutyryl S acyl carrier proteins to form crotonyl S acyl carrier proteins and some amount of water molecules. And this third stage is known as again reduction. The reduction sequence is repeated to give 6 carbon unit, 8 carbon unit, 10 carbon units and so on. Finally, the most important fatty acids with 16 carbon and 18 carbon units are produced. When the required chain length has been attained, the acyl group is transferred to HS group of coenzyme A. Finally, the coenzyme A derivatives of fatty acids are formed, which are used up in fat synthesis. The above mentioned reaction occurs in the cytosol at the endoplasmic reticulum. These reactions are catalyzed by a multi-enzyme complex called fatty acid synthetase complex. The enzyme complex probably contains 6 enzymes, each catalyzing one step. The overall generalized reaction or equation for the synthesis of palmitate is summarized as follows. 8 acetyl coenzyme A combines with 4 ATP plus 4 NADPH. It gives 14 NADP, 8 coenzyme A plus 6 molecules of water plus 7 ATP and 7 IP. 7 ADP adenosine diphosphate plus 7 IP inorganic phosphate. Formation of glycerol. The glycerol is most probably synthesized from glyceraldehyde 3-phosphate, an intermediate formed during glycolysis. The steps involved are as follows. Glyceraldehyde 3-phosphate is first of all dephosphorylated to glyceraldehyde in presence of enzyme phosphoglycerate phosphatase. Glyceraldehyde 3-phosphate is converted into glyceraldehyde and inorganic phosphate. The glyceraldehyde is reduced to glycerol in presence of enzyme aldose reductase. Reduced NADP, that means NADPH2, get oxidized in the reaction. And number 3 is combination of fatty acids and glycerol to form fats. Neutral fats, that means triglycerol, are synthesized by esterification of molecules of glycerol with 3 molecules of fatty acids. All the 3 molecules of fatty acids may be the same or different. This process occurs primarily on the surface of endoplasmic reticulum in the cytoplasm. In general, the biosynthesis of triglycerol in higher plants resembles that in animal tissues. The key precursor of triglycerol, that means phosphatic acid, may either be formed from dihydroxyacetone phosphate or from glyceraldehyde 3-phosphate. Steps involved in the synthesis of triglycerol are as follows. In first of all, the fatty acids are converted to their acyl coenzyme A derivatives in presence of ATP and coenzyme fatty acyl coenzyme A synthetase. This conversion occurs in the microsomes. That means glycerol combines with 3 fatty acids to form glycerol fatty acids coenzyme A. Number 2, glycerol is phosphorylated to glycerol 3-phosphate in presence of ATP and enzyme glycerolase. Glycerol combine with ATP to form glycerol 3-phosphate and ADP, adenosine diphosphate. Number 3, glycerol 3-phosphate combines with fatty acid coenzyme A to form lysophosphatidic acid in presence of enzyme glycerol phosphate acyltransferases. The molecules of coenzyme is released. See, the glycerol 3-phosphate combine with fatty acid coenzyme A to form lysophosphatidic acid and coenzyme A is released during the reaction. Number 4th reaction is lysophosphatidic acid combines with second molecule of fatty acid coenzyme A to form phosphatidic acid in presence of enzyme 1-acyl glycerol phosphate acyltransferases. See, the lysophosphatidic acid combined with fatty acid coenzyme A to form phosphatidic acid, and during this process coenzyme A is released. 5th reaction is the phosphate residue is cleared from phosphatidic acid to form a diacyl glycerol in presence of enzyme phosphatidate phosphohydrolase. Phosphatidic acid is converted into diacyl glycerol and inorganic phosphate is formed during this reaction. 6th reaction is finally the third hydroxyl group of diglyceride is esterified with fatty acid coenzyme A and produces a triacyl glycerol, that means neutral fats, in presence of enzyme acyltransferases. Diacyl glycerol combined with fatty acid coenzyme A to form triacyl glycerol and coenzyme A is released during this process. Thank you.